Secretary NTA, and my own long experience in this cluster. So I would like to know from you that anybody who is from agricultural entomology or entomology, and breeding, silkworm, any animal breeding or plant breeding, plant breeding, and GKVK people, how many? You all must from GKVK. It's famous. It's becoming now worldwide famous. GKVK for sericulture. I have visited that place. Nice sericulture. So I'm starting the conservation and breeding. So this, you know, conservation and breeding are interconnected economic issues. You know, now their era is of bioeconomy is coming. That you people are biological background. You know, bioeconomy is a revolution.
Hello. Hello. You are
Hearing my no need, I think.
Need है.
okay. Hello. Okay.
Yes, sir.
Okay. That with you. So, these are natural breeding is a part of in situ conservation. It is okay. You may get it. And the artificial breeding is a part of ex situ conservation. You know in situ and ex situ. Now, what is conservation? To protect the habitat ecosystem for a Manita from extinction, protect erosion of biotic interaction. What is biotic interaction? You know very well that for survival of one of the species, there are operational activity of other life also there. Use of interdisciplinary science, economics, natural resource management to achieve the SDG through ESG that you know. Process of developing, improving, maintaining races through plant mating and selection. Objective of enhancing desirable traits such as high open yield, superior silk quality, disease resistance, adaptability to a specific environmental condition. Now, you know the global distribution of Antheria milita. You just see here, Southeast Asian country, that is the part of Oriental geographical zone. There, Antheria species are distributed. It is not only in India, it is everywhere, but they are not commercialized. Somewhere it is commercialized. In Africa, South Africa also, in other places also. Now, types of silk in India, you are knowing this tropical tussar, oak tussar, eri and muga. Muga is also called golden silk, and eri is a cottony silk. How it is called cottony silk? Have you seen eri? How it is called cotton like silk? Because it is spun. It is not come out as a real one. It is spun like a cotton. And you might be knowing that you might have heard Dhaka ka malmal. This is that eri silk is the Dhaka ka malmal. Malmal means cotton, but it is not cotton. Dhaka ka malmal, which used to come in a matches box. सुने होंगे ना कि जिसके पूरे फैब्रिक जो हैं वो उसमें आ जाते थे. Such thinness it may come in the eri. Now come to the tropical tussar, eri. These are all, and these are Milita, Asama, Royally. You know, Antheria Milita chromosome number 31 N, Asama 15, Royally and Proily how much? 31. Parnai 49, perhaps I will show you. Yamamai 31. So one is silk forest ecosystem, which type of forest ecosystem they are surviving, that is there. And one is silkworm, what are their host plant? Antheria proily, Quercus serrata, Antheria asama, Persea pomsina, Litaceae monophylla. Temperate oak tussar, these are the cocoon. Now this is the distribution. Now this is the ecorace you are seeing here. In the Jharkhand area, majority of the ecoraces are there. Now I am explaining you, this is very important. Actually regarding tussar, I am coming to tussar. Tropical tussar, you have seen it is distributed in Andhra also, it
is in Maharashtra also, it is in Chhattisgarh also, it is in Odisha also, it is in Jharkhand. But the seed industry, actually you people have joined just or have posted somewhere? Not posted anymore. Then BSMTC, you know, basic seed multiplication and training center. RTR is Regional Tasar Research Station. Okay, it has become RSIs now because involving other also. Just to shrink the whole organization, just utilization of the maximum work by one station. REC, Research Extension Center. So, but the thing is that all BSMTC works on DABA. DABA is a course on which the whole seed industry works. Seed industry means BSMTC. Your Tasar productivity are the outcome of BSMTC only, because they produce the DFL, the seed which is given to the, but actually BSMTC, why it is called basic seed material. Basic means the first, I will come to the, because you people are new, so I will first show you this one, then only you can understand. Then I will come to that. Crop cycle and seed organization. Actually, the first drainage is the outcome of preserved cocoon. First drainage, that is called basic drainage. It is outcome of the preserved cocoons. That means diaposing cocoon. So it is. You can see now. You can see now. That is. So first drainage and first rearing, second drainage, second rearing. 4 events are there in 1 year. So that is the crop cycle. First drainage is the basic drainage, occurs on preserved cocoons, June, July. Now first rearing, first crop, basic rearing or ampatia occurs on the DFLs of P2 basic drainage, July, August. Second drainage, commercial and nucleus occurs simultaneously. Now, elite seed, nucleus seed, basic seed, commercial seed. There are 4 layers of seed quality. E4 is the elite seed, high quality geopebrin with very high yield and productive traits. That is P4. And P3 is nucleus. P2 is basic. P1 is commercial. That some parts GKBK people might have heard, learned in the sericulture. You know now, in Mulberry. So this P4, P3, P2 and P1. But we have bivoltine area, and now come to the Tasar has 2, bivoltine and trivoltine. But trivoltine is not too much. It is in the hot places. That is Chhattisgarh area. Trivoltine Chhattisgarh area means 3 crops. But it's not constituting the major. The major contributions comes from the bivoltine. The first crop which occurs in July, August, the crop means rearing, and the drainage occurs prior to that, that is June, July. And second drainage, commercial and nucleus, means 2 direction. It takes 2 direction. Why? Commercial, because there are 2 only now. So one side it goes to commercial, means the seeds will be given to the farmers for cocoon formation and selling. It will not be taken into further recycling, further in the your multiplication process. And second rearing is very, very important rearing, commercial and nucleus, that is P3 rearing, that is called a nucleus rearing, simultaneous from cocoon of second drainage. So now the first drainage takes place during June, July, that is the hot climate. Just first shower of rain that gives the emergence of Antheria mylitta moths. You know this Antheria mylitta, the large sized moth, and they outcome of the larva which feeds polyphagus on various host plant. But the primary are the Arjun and Asan for DABA. But for ecoraces,
sal is there, and other. That is why polyphagous. Secondary food plants also. Actually, Tassar culture. Uh, so you have understood this. This slide you have understood. Any problem in this? Then I will come to the origin of Tassar silk industry. You understand it? Any problem? Because you people also know. Mulberry people, GKBK people might have understood. Entomology people. Why I am telling entomology people are so much required and they should take interest, because Tassar is exclusively more important for the entomologist. They should understand the insect. The entomologist might be knowing insect chemical ecology, ICE. Insect host plant interaction. There is a number of words. Hari Anant Katha Aginanta. There are a number of words. ICE, insect chemical ecology. It's a very vast area, and you have to do. You can come up with the publication. You people posted either in the research or in the seed products or on the regional agriculture research station or extension center. Please stick to the experimental work and publication, because now it has come to the FCS. We are the people, we have driven it to bring the FCS. I was the first instrumental in bringing this FCS during 2007. I will tell you the story, how we done it. So this, your publication from the BSNPC should also come up, from RSRS, REC also. And you should do ICE, insect chemical ecology. There are two pheromone, caromone and cynomone. You know cynomone? What is cynomone? It attracts the larvae to the host plant. And caromone, larvae to larvae. They communicate, larvae to larvae. Larvae, moth coming to one host plant, that is caromone. They are attracted, and the pheromone, sex pheromone, the male comes from outside to the female. I have done it experimentally in the forest area. I will show you, you will see that photograph. Just hanging the female moth on the shoot of the forest plant, the males used to come from the adjoining area. But you will tell how it comes. It always comes from some den. It should come from certain habitat now, where their multiplication were there. Now one thing you tell, how silk production can be enhanced? Just throw your mind. Silk production कैसे बढ़ाया जा सकता है? Means कोकून प्रोड्यूस करके ना, कोकून को ज्यादा प्रोड्यूस करके, ज्यादा प्रोड्यूस कौन करेगा? Who will do? Farmers. So you know the very existence of your silk industry, that comes due to farmer. Always remember, farmer is your god. Farmer is your god for every sericulture. Farmer is your god. He has invented, you know, this tropical tassar. Why I am telling, Daba belongs to Simbhum. Why I am telling that, I will come. Now come to that origin point, that one. That's right. Origin of Tassar by Itna D in Saranda Simbhum forest corridor upon natural breeding, perpetuation on Soria flora. Soria, you know, Sal forest is distributed all throughout the central India, in the Deccan Plateau, the South Central Plateau, throughout. You know that Saranda, you know Simlipal in Odisha, you know Dandakaranya in the south, which is the joining point of Andhra and Maharashtra. So white soil Daba is a parent of landrace Daba, perpetuated in and continuing in Econicage. Downstream on Asan and Arjun, tribals of Chotanagpur practiced since millions years and invented in form of landrace Daba. Countrywide adopted in base is the base of Tassar silk industry existence. Whereas those which remains wild only feed on Soria, multiply self as wild to wild cocoon. Wild to wild, wild to semi-wild, that I will come. Now semi-wild feeds on various host plant. You read there and I am explaining you. Actually what happens, one way the Antheria marita was having their population in Jharkhand area. You know that Jharkhand is the leading producer of tasar. Its origin took place in Jharkhand itself, in Sirbhum, West Sirbhum. So what happened there? Since time immemorial, means thousands of years ago, tribals of Chotanagpur brought the cocoon and just bred them. And the cocoon comes out of their rearing, they sold to the market. Because the reelers, which are from Banaras area, reelers or Bhagalpur area or Champa area or wherever there the as manual reelers were there, they used to collect cocoon, they used to purchase cocoon in the heart, weekly heart of Jharkhand. So this has pushed them to collect more and more cocoon and breed them, and in this way successively this has transformed this wild daba into ruling daba, which formed the base of Tasar silk industry. Actually this institute was opened in Chaibasa. Chaibasa is the capital of Sirbhum, West Sirbhum. So this was in 1959, this was working there, and later on it has been established in 64 here, CTRNTI. So it was working there only. So this all BSMTCs has been formed by taking the daba from Chaibasa to different places, Madhupur, Kharsawa, or Chhattisgarh area, Boydadar, Raipur, all other places, the daba has been supplied there, daba. So daba formed the base of Tasar silk industry. Means this is a manually handled eco-race. Manually handled. It's a semi-wild, you can say. Semi-domesticated. It is transforming towards semi-domesticated. Means its grainage is done out, rearing done in the block plantation for the daba. But for the eco-race, which I have shown there are wild, semi-wild, two types. Wild and semi-wild. Wild means if cocoon you have seen on the shoots of the sal, Surya robusta, then you are saying that moths are coming up and they are mating in nature. So this is from the sanctuary where you are not interfering. But what is the difference between the zoo, park and sanctuary? There is a difference now, you know. Zoo and park, you are doing manually, means interfering something. But in sanctuary, you are not disturbing anything. So just like that, Tasar Silk Sanctuary, this Saranda Sirbhum Forest Corridor is working as a sanctuary. So that is why I have told you that when I just hang the female moth, the male came from kilometers due to pheromone. They attract and come and mate. So this was there earlier also. This was a natural event which we have observed and it is doing. And this, so those daba which you are perpetuating on Soria, they are called wild daba. Okay? If that is, I am permitting wild to wild transformation, means I am not interfering anything, then it's a complete natural, it's a institute, it's complete natural. There it is zero pebrin, no pebrin is there. Now I am coming to the pebrin point. Pebrin is the dangerous and the most crucial and the challenging and the very interfering factor in breeding also. Breeding what in here? Also breeding. Pebrin is the main problem. I am telling you. Pebrin, if anybody come out with the solution of pebrin, he may be awarded the Louis Pasteur Award. Really, it's there. And for this wild, this tasar, where it is a outside rearing practices there is many chances of exposure and the incidence of pebrin every time this happens, because the pockets, rearing pockets, once you are repeatedly rearing, it becomes sometime incidence of the pebrin comes and the pocket becomes infected. You are not able to reduce the percentage of pebrin. I will also tell you how pebrin percentage can be reduced. Now come to this, how this cash crop of tribal farmer of Tasar states livelihood employability earning, this daba is doing for this. Cocoon collected from sal by forest dweller farmers sold in local hart. You know that cocoons, big size cocoons comes and I have brought it. निधि मेरे बैग में के पास टेबल पर 1.5 to 2 or actually in that life cycle also I should tell that Ampatia cocoon cell thinness will be very thin, but in commercial or the nucleus rearing the cocoon will be thick. Why? Because the larval span is long, means larvae takes more time to eat and the cell become thick, means high silk is there, long number of filament. It is about 1600 meter filament long. So you see this very big size wild dava, this big size. So along with the pupa, their weight is coming to 18 gram or 20 gram like that. So very big. So why to resort for this? Just to have, if that is in the homeland of tropical tussar, that is West Singhbhum, there are abundance of such cocoons. So what we have to do for breeding, was posted to open P4 tussar breeding station in Chakradharpur. There were no any advices from the institute. I just myself created the thing, how to do, created the thing. Because when I was posted there, state government was doing such a stock rearing. So if everywhere pebrine will be there, you will not be able to continue your stock. It's a challenge for the breeding. It's a great challenge for the breeding. So that's why you have heard that many times I am telling wild dava, wild dava. Even this institute scientists have also become very much allergic to that. Why I am telling wild dava? It is only because of the fact that I have told you, if natural in situ perpetuation is there, no intervening by the human, then it's no pebrine, zero pebrine. So I have got the source as zero pebrine in the wild dava. So I have brought it and just reared. Their mating percentage are not such as in ruling dava, because ruling dava become adopted. It has become semi-domesticated, so the percentage of mating are high. In the BSMTC or here in experimental also, when you will do, you will see mating percentage 70 or 80%. But in wild one which I have brought and done, I have got 40% mating, because it's a behavioral entomology. If you are intervening, its biophysiology, bioprocess become intervened. So it is not doing the complete as in the domesticated. You are saying that mulberry, domesticated everything, its egg number, egg size very small, egg number 600, 1000 of years, about 10,000 of years it has been domesticated. But no doubt this tussar is also very old, but why not domesticated? Why not domesticated? Again comes the role of farmer. Farmers are not being instructed to do its domestication. Sometime in this institute, so many times domestic means indoor rearing process has been adopted, but not successful, because I am telling you, there is a wild to semi-wild to semi-domesticated to domesticated. There are 4 stages: wild, semi-wild, semi-domesticated, domesticated. So this phase of successive transformation, this takes 1000 of years. So that's why now we have nothing other option, just resort to just do the rearing out of this wild dava to take the zero pebrine stock and then rear it in zero pebrine pocket. Always remember, it is not that the silkworm carry the pebrine. Wherever the pebrinized silkworm is reared, that host plant become infected with pebrine. So that is why there is a usual practice that just change the pocket. Sometimes just search the new pocket. So Purjuli as the new pocket we have identified there. And for Nidhi, I am of the opinion that for any breeding program, whenever you are convincing any members you tell that I will appoint one GRF for Kurjuli, and there we can monitor the throughout the breeding process, and there we can get the selection breeding, recurrent selection lines or whatever, because that is the area which is free from peppering. So farmers are giving the yields of 90 cocoon, 100 cocoon, 110 cocoon like that. It is only because of the fact that the host plants are nutritious, salubrious, and I will show some photo also that Kurjuli area. So every time in Tasar, you will face in future, and if you will post it to the BSMTC or REC or whatever area, you will face that area which are becoming for the indication of peppering, that you will be unable to send rear there, and why it happens, that I will explain you. But first come, these are simply distribution. Achha, now about the wild dava, I will give you this pen drive. You can take these all details. Why wild dava may be the oldest ecorace. The origin of homeland of Mylitta is Kolhan Saranda. Only dava is countrywide adopted, no other ecoraces. They are regional. The seed organization operates on dava. The ex situ multiplication of wild dava is 60 to 70%. Means wild dava can do 60 to 70% ex situ multiplication, whereas other ecoraces are not doing like that. Highly amenable and subject to large scale multiplication. Evolved as ruling dava by the age old practice by tribals. Dava has maximum number of SNP. Prabhu, SNP alleles are high in dava, wild dava. Major and minor ecoraces are accumulated in these zones. Now, one thing is there which I have told you, that male used to comes. Actually, you, how many days you are here? कितने दिनों से हो यहाँ? Rearing field गए हो?
Yes, sir.
So this is a systematic plantation. You will find in BSMTC, no doubt, in some PPC also, in REC also. But farmers do not have this type of plantation. This is another challenge I am telling you after peppering. After peppering, this is the most dangerous. Dangerous means farmers are doing rearing in the unsystematic tall plants. You see, tall plants. So there are so many losses due to the pest predators, and they are damaging their yield. So their yield are coming very less. Only some dedicated farmers having zero peppering stock or good stock, which is given by the BSMTC or the PPCs, they are doing good performance. That is why I am very sorry to tell that the production which has increased during the last 10 or 15 years, that was 27,000 metric tons, it has reduced to 1,500 now in Jharkhand. In Tasar as a whole, taking all other Chhattisgarh, it is 1,589 metric tons. So now that you people have on your head the challenges to meet and increase it if you are posted in the Tasar. Now, what is the problem? Farmers are putting, not by doing any type of scientific process, just taking the DFL in the cotton box, hanging it on the tree. So you see many of the worms which come out die there, self, and then they just crawl and go to the different branches. They are even not maintaining the chalky stage. You know, chalky stage, those GKVK people might be understanding, means those plants where salubrious and new foliage are there, soft foliage are there, young foliage are there for the first and second stage, because their mandibles are not grown. That is why the soft plants, there should be a chalky level, that means up to 4 feet. Host plants should be there, and there should be nylon net, so that they should be protected from the outer predators. You know, Gandhi bug, they beat as a predator, that beat that there, and the Hymenopteran, they just pick up so many small and they carry on. So these are all the losses the farmers are getting. So many losses are there due to they do not have systematic plantation. They have large plantation. Now again, these are the losses, but there is a plus, something plus you can say. The male which I have told you that used to come, That is due to the fact that farmers are harvesting the cocoon up to a similar some heights, but they are leaving all those cocoons which are on the top. So those cocoon, the moth emerge and they just hovers all along the top part of the forest area. So there is formed the natural reservoir of the kasar silkworm all around the forest area. Not only that, I am telling Sananda Simon, even the BSMTC who are doing for their own multiplication of DFS, they have the systematic plantation, but in the commercial they are giving it to the farmers. Farmers do not have. So farmers have large plantation unsystematic and they are putting there. So almost all the BSMTC area will have this type of just moving moths which are emerging out of their own population on the top. And that forms, now there is a challenge. If that stock is natural, good, zero peprin, without peprin, it's okay. But if it is peprinized, then it's making a very dangerous signal, very dangerous. That is why when I opened P4 in 2010, I have met with that. Earlier I was knowing that the eco-races do not have the peprin, zero. But when I have seen, I have told you now, the state government, just to achieve some target, they have put just without any good quality, without any testing, so many of the cocoon in their PPC. 200 number of PPCs have been made and they have put there. Now, untested means the peprinized population has emerged and it has just encroached all the forest area. So, that peprin is causing a cascading effect. Cascading effect, means if it is 2%, 4%, it will multiply, it will go to the 10%, 20%, 30%, and it will blast, complete blast of the peprin and there will be a loss, great loss. So that is why earlier I have told you potential of wildflower, same thing. So now there is conservation types of ecoraces, wild, Rayleigh model, wildflower, Laria. Sarihan, Bhandara, local. I am giving you three which I have told you, wild, semi-wild and captive. Daba is a captive breed, okay? Captive breed. You can do that rearing by handling, manual handling. But wild and semi-wild, you see here, about Rayleigh I am telling you one story. It's a lively story. The farmers of the Chhattisgarh, they used to cordon the whole of the forest by putting cocoon. They do not disturb. From there only the Rayleigh has come. They were the natural scientists. They were not disturbing that complete pocket in the Chhattisgarh area, and in that area they were putting and just keeping it for 3 to 4 years, and then they collect from the forest. So there, that is complete wild. Rayleigh was a complete wild. Means if you are not touching any part, any developmental stages, that is complete wild. But if you are touching the stage, it is semi-wild. Why? Laria also in Jharkhand, once Director of Sericulture Dhirendra Kumar was there, he was very much ambitious of just doing that Laria. Human interference was there, Laria cocoon. But why Laria is a small size and big size both? Why it happens? Throughout the year, there is a climatic cycle, seasonal cycle, there are altitudinal impact is there. So if you are collecting either one Rayleigh from one forest, big size, you are doing grainage, that means what? You are interfering by manual handling now. If you are doing manual handling and then you are putting that larvae on the host plant, which Sal, Surya, their size becomes small. And that correlates with the unfavorable season also. But when favorable season is there, rainy season, the size become big. So this type of cyclic transformation occurs in the area also. And but some of the, that is Sarihan, which is cordoned in that area, it is confined to one area, that is your yeah, Dumka area. Some forest area are there. What semi-wild means what? Drainage they are doing manually, but rearing is done naturally. Means they are putting the DFL out of the Sarihan on various host plants, not only Soria, other host plant also. That is why I have told polyphagous. Semi-wild, polyphagous means other host plants are also, there they are feeding. So if there are so much variation of the host plant, automatically the cocoon variation will be there. So the variations are there in the ecoreces. Why that really were there, and up to some extent I can tell what I collected wild dava from the natural reservoir. So there you can, that we can say as the truly wild. But other whom we are collecting, putting the cocoon for the drainage, then what? It is a human interference. Putting drainage, you are interfering. Maybe that you do not do so much of the interference just like dava. You just garland it. I will show you. There are so many methods of conservation. Just have a pagoda type structure where the garlands are hanged, and you have not to interfere anything. So what happens there? Their natural cynomonic interaction occurs. Why conservation are done in the habitat of the forest? Because cynomon and pheromone interaction occurs. In the host plant, due to the host plant attraction, there the cynomon emitted from the host plant that attracts the larvae, and the pheromone emitted by the larvae moth together for their own communication. So they are semiochemicals, communications are there. So this is the conservation one. Now, cocoon source pockets. I have told you now that the plants are variable. So those plants type I have told written here. Block, systematic plantation, for whom this human manual intervention, unsystematic aggregated, unsystematic distantly located, forest pocket on hills, forest canopy in foothills, semi-forest patches, rice field patches, upland, lowland. Variety of host plants are there. Variety of host plants are there. So now you see how the variation takes place in Tassar silkworm. Now at this place I will explain you one thing. Ampatia, or the first rearing, usually it falls during the rice crop. धान की खेती होती है उसी वक्त ही होता है, तो farmers do not opt for this. So that is why there is a lack of linkage of the seed. What I have seen in Hadgamaria is a very good pocket for the Ampatia. You will see there are so many host plants all along the interconnection of the rice field. In between the division of the rice field, there are all host plants are there. So when they are doing the paddy crop, they are doing rearing also. The fecal matter which falls on in this, that is also a fertilizer for them. So there the first crop is excellent, Ampatia, in Hadgamaria area, that I have seen. So that fill the demand in the second, because it's a linkage now. So if the Ampatia or the first crop will not be successful, will not be opted by many farmers, then how the next demand will be filled up? So that is why there is another, I have told you the most important challenge, peppering. Second, plant unsystematic. Third is Ampatia is lacking. So what we can resort for? We can resort for all those farmers which do not have their plantation in the rice field area, they can do. There, if you do the plantation in coming era, you people can do it, come in the Tassar field. You do that plantation, then the Ampatia crop will be opted. Then the demand in the second crop, that is the commercial or the nucleus, that will be filled up. So this is the third most important challenges which you will come over. Now how ecoraces evolve. These are some of the details given. Distributed the outbreeding population in nature. Disruptive selection operates to create population diversity. Geographical barrier creates reproductive isolation. Splitting of population of species occurs, so though mate but no sexes transform into regional population equilibrium. Insect plant polyphagy result in colonization on host plant. Genetic polyforming facilitates to stabilize in a fresh eco-environment. Restriction of gene flow, population flow successively adjust to fitness in particular ecozone. Based on population density, area of availability and foraging zone can be characterized as major and minor. Based on insect behavior, availability and food plant, these are characterized as wild, semi-wild and captive. Getting my points here? Progress measures. Progress measures, RTRS has been established for ecoraces. And Rayleigh model, Serian, Bhandara, Andhra local are the very existing RSRS now named, but they were RTRS for Andhra local, RTRS for Bhandara, RTRS for Rayleigh, RTRS for model. There are center which has been established right from during the establishment of the BSMTC. Just after that, BSMTC will first installed in Central Tertiary Research Station by under Central Support. BSMTC was the first thing which has brought up the productivity, and then it has been found that regional ecoraces are there, so that's why they installed the RTRSs. So there now you see in the same state there are regional ecoraces, there are BSMTC. Now you will come to another challenge, fourth one. Some farmers will prefer Daba only because their yield will be more, then ecoraces will be reducing. So now this is your fourth challenge coming era, you people new coming to the professor. You just divide some of the farmers to the Daba and to the ecoraces. Then the productivity will come up. Only on Daba, you cannot just push up the productivity on ecoraces side also. So this is the fourth one, that your training towards the ecoraces to the farmers, that is to be taken up, and the BSMTC is operating in its own way, that will do. So further technology are being refined and fine-tuned on Lairia, Modia and Jatadaba. RTRS now RSSs were established during 1986 and Warangal in 1992, and subsequently the developed technology were in practice unit. US involvement are also there, but need to be strengthened. Why need conservation? That you know. Massive depletion of tertiary forest flora. Farmers, forest dwellers, they cut the forest for their fuel and for selling in the market. Threat of complete genetic erosion, irrational collection marketing of nature-grown cocoons, deforestation for fuel, livelihood earning by tribals, increase in anthropogenic activities, unawareness in the tribal or restriction of collection, extensive losses due to parasite predators, climate attacks by heavy rains, untested disease seeds thrive to the core buffer area. This is the last point which I have told you just now. This is another challenge coming up. Now the source agencies, I have put same thing, CSB, CTRNTI, P4, elite seed, CSB, BTSSO, P3, nuclear seed, quota, CSB, BTSSO, P2, basic seeds, all BSMTCs, DOS, all PPC, P1, commercial seed, BSPU, CSPU, basic seed production unit, commercial seed production unit, nuclear seed production unit, nuclear seed rearer, basic seed rearer, commercial rearer. These are the structural system of the seed system. CSS, scheme based sources, rest all basic and commercial. And in ecorace side, CTRT, RTRS, DOS, PPC of ecorace zone, adopted ecorace producers rearer. You are getting the point? This side Daba base, this side ecorace base. This is the canopy Muga eco zones. Now coming to the Jharkhand Meka eco zone, Wild Daba, number 1, these are the places. Then this Modal, Sukinda, Sukindagarh, Nalia, Simlipal, Amarkot, there are so many minor ecoraces. Now come Chhattisgarh. Raleigh is the main Bastar district, and so many other are also there. Bhandara in the Maharashtra, Andhra local, Adilabad, Karimnagar, Tira, Purulia. So these are again the list of the different states having the districts involved with Tasar. Conservation technologies. So the drainage preservation in nature, cocoon garland system, horse tree net cover technique, pagoda device technology, wire mesh feather technology, and the proliferation in nature, release of mated moths, release of unmated moths, release of egg in leaf cups, release of chalky worms, cordoning of forest patch, periodic checks on conservation. Ex situ conservation technologies, MGH, offsite, disease rejection of unfits, rearing in natural tree forest, rearing of host plant stasis, unsystematic forest, semi-forest, rice field dividers. This is where you are knowing the core gene pool reservoir is the pure, and the buffer zone in the conservation, what is usually done, we put the population in the buffer zone. So when they multiply, they can go outer side, they can go inner side. If they go inner side, they form the gene pool accumulation. Okay, you leave it, don't touch. You leave it to multiply. But the outer side, if it goes in the peripheral area, that you permit for collection. So this is the core, buffer, and peripheral. What this is the base of concept is that the core area should be left for reservoir creation of the high yielding, high quality population, natural population, in situ population, and don't collect from that gene pool reservoir, the core area. You just put garlanding in the buffer area. The population which will multiply, they will automatically go to the inner also, they will go to the outer also. Those which will go inner, you leave it. Those which go to the outer, you permit for collection. Then there remains a 60-40, means 60% you are collecting, 40% you are permitting to remain there. Then their self-perpetuation will enrich the population. So this is the main fact. So care after proliferation, periodic cordoning, periodic checks on conservative exploration of new pockets. Three things can be taken up every time for the conservation people. And these are the technologies, photograph of the technologies. You see, leaf cups, clipping of leaf cup, pagoda shrub mate. Research and taken long-term strategy. What I have told you that the entomologist or the all sericulturist can do long-term research on this also. Bioprospective of potential econogen, core buffer, peripheral. Formation of conservation farmer cluster, niche by regular survey. Baseline survey of population dynamics, cost, plant and season. Recording of reproductive cycle, crop cycle, qualitative, quantitative. Insect host plant interaction, ICE, what I have told you, cinnamon and pheromone. Bioprocessing uses CSV NESAC, that Dr. Prabhu is doing, CSV NESAC collaboration outcome. Insect nutrigenomics on RNA, proteomics and metabolomics. Agroforestry and joining allied culture to create umbrella effect. To develop conservation eco-park, Tasar eco sanctuary, integrating bioeconomic approach on natural resource. Umbrella effect, you know? Anybody? Ecology?
By conservation of the one species, other species are protected in that area.
Very good, very good. So this is a you can say community development of the biomass, bionomics and bionom, the all increases there, and then comes agroforestry, allied culture you can do. Acha, insect nutrigenomics, RNA, proteomics and metabolomics. You know, proteomics is now the era. DNA is obsolete nowadays. Only the proteomics that gives you the indication for what is the biomarker. Metabolomics is also there. In case of this, you know, sal, there are so much feeding deterrents in the sal. So only the wild larvae can feed. So you can identify the biochemistry among you, which factor is working as a biomarker, and you can identify the biomarker which is permitting them for the feeding on that. So insect nutrigenomics and nutrimetabolomics, you know very well. If biochemists are there. Now extending CSB DOF coordination. So assessment population growth data by forming use of Forest Department VSS, One Suraksha Samiti, Dasar Vikas Samiti, Dheeraj Forest Committee, Joint Forest Management Committee, organize secure sustainable earning livelihood prospect, organize gap filling plantation around the core buffer peripheral zone. Formulation of conservation scheme under CS. हाँ, एक important point है मैं बताता हूँ. जिनकी the fact is that you are the upcoming scientist. You can just have proposals. Taking the help that there have been made a model of cost structure of modal Rayleigh area. Based on that, from the developmental scheme of Central Silk Board, there are developmental schemes are approved under Central Sector Scheme. That is mostly given to the state. But those people who are in RTRS, they can ask for that CSS together with a state. They should formulate certain project for their implementation together with under CSS, Central Sector Scheme. Central Sector Scheme, there are organized project in which you put the cost of laborer, farmer, cost of plant you are taking up in collaboration with the Forest Department. So with the DOF means Department of Forest and DOS means Department of Sericulture, Directorate of Sericulture. So just have a collaboration and take the Central Sector Scheme counted under this conservation counted under Central Sector Scheme. Till date it has not been made. अभी तक नहीं हुआ है. ये हो जाए तो अच्छा होगा. That's why this is the fifth and the sixth point. To collaborate with University College High School near to conservation zone for orientation of importance of fertility, biodiversity and culture conservation. These are the some collections and forest area. Sensitization program for local forest, semi-forest dweller, JFM city with RFO, DFO to construct national working plan of core buffer periodic zone forest. Support from market and Mahajan and backtracking of the collection process from Econis. देखो ये बताए थे 6 point है. Whenever you will go to the eco-race zone, you will be blind. You will not know the eco-races where it is. Where from you will take. कोई eco-race area में गए बस करने. तुमको कोकुन चाहिए. You want कोकुन. Where from you will take. Market. वीकली हाट में महाजन के पास फार्मर्स यूज टू सेल देम. फ्रॉम देयर इट इज कलेक्टेड ऑनली. फ्रॉम देयर इट इज. When you will go to the RTRS area and the state coordination, you will find that this is the machinery working. Now you in the research paper and the projects you formulate so many things, so many systematic. But the thing is that Mahajans are working with the farmers. Farmers are collecting for the forest area, deep diving into the forest, and they collecting that and they are giving it to the Mahajan. And Mahajan means why we are telling Mahajan? They are putting them preserved. And when there is a demand, high demand, they sell it in the cost of 12 rupees per coupon, like that. So that is on the way out. So now there is exploitation of the farmer and exploitation of the scientific sericulture industry also. Why? They are putting all the cocoon which has come from the farmers in one place. So there you have to play your role to take direct linkage with the farmer, scientist or the technical staff to the farmer and not through the Mahajan. But the Mahajan are operating there. Why? Because there is a very thick cocoon. This is a very costly because the silk will come more. So that is why there also this is the 6th point here. Keep in your mind. The Mahajan's quantum of collection in silk constitutes overall eco-race collection which add to the state culture silk production. Overlapping crop result in scanty cocoon population, but when huge number of cocoon are kept there are maximum chances of same day coupling. Also to increase same periodic close period crop cycle, the male are attracted from far towards female to mate by pheromone. That is there. This result in build-up of abundant population from parent collected and hanged cocoons for use in next cycle in favorable period to inoculate in different sub-camps under main camp. Thus proliferating population migrate towards both sides, core and peripheral, and hence it acts as a buffer zone. Out of magnanimous of vegetation population, only 60% should be harvested and 40-30% should be left in the eponage to thrive. Operation wild to wild perpetuation, suo moto in core area. That I have told you about the rally and wild daba. Semi-wild in situ, ex situ in buffer zone. Here drainage is indoor, but rearing is spread among polyphagy. Cocoon collected from semi-forest zone, buffer zone. Collected cocoon are subjected to drainage either in situ or ex situ. DFS eggs are supplied to local farmers adopted here for rearing along the forest. There is transformation from larvae to cocoon, semi-wild to semi-wild. Cocoon formation and maturation have uniform period of crop cycle. Emergence occurs on uniform manner, negligible overlapping. Crop period cycle runs throughout the year. A strict compliance on zero tapered status to be followed. ये कॉस्ट बेनिफिट रेशियो का एक कलेक्शन है, कैलकुलेशन. नाउ impact and benefits of conservation are there. Why the need of biodiversity conservation? There are so many factors. Why the need of conservation of wild lava? High value of wild lava का conservation इसलिए भी जरूरी है because it is linked to the ruling lava. So its progenitor parent is there, so we have to keep it. It's by its conservation. Breeding. Now coming to the breeding types, you know, inbreeding, crossbreeding, selection breeding, recurrent selection breeding, marker assisted breeding. Inbreeding, cousins, double cousins, half cousins, you know, seed breeding, sister brother mating, very close mating, and then half cousins, double cousins, details are given here. Crossbreeding, crisscross and triple crossing. Advantages of crossbreeding, you know, just to eliminate inbreeding depression, we have to do crossbreeding. Heterosis is a phenomenon in which hybrids of genetically dissimilar parents are crossed so as to increase vigor over mid parent. So one example is given here. You can calculate, a calculation is also given, P1, P2, F1, CV, like that, calculation of heterosis percentage that we used to give. Now challenges in breeding. Non-compliance to maintain zero papering, not focusing on DABA based breeding, not focusing on DABA based breeding. Why I am telling? During the last 25-30 years, there were so many papers you might see here in the annual report, etc., doing interracial process, but they do not succeed. Because if you are doing DABA with Rayleigh, DABA with Syrian, DABA with Modal, not finding. Why? Tell me why? Not finding sustainable, not finding a sustaining effect. Why?
Original parent is the double.
DABA has original parents. DABA. It has become a eco-race or a variety, you can say DABA. It has become a variety which you are developing in different BSMTC as the regional variety. Because BSMTC are everywhere. In Andhra Pradesh, in Maharashtra, in Chhattisgarh, there are 4 or 5 BSMTC are there. In Jharkhand, 4 or 5 are there. In Odisha, 4 or 5 are there. So the ruling DABA is multiplied. So you have to restrict your breeding in the DABA only. There should not be any interracial means DABA to Rayleigh, DABA to Medal, DABA to Syrian. It will sow once because they have their parent Antheria mylitta tree. Prabhu, genetically or the molecular genetically, there are so many identities among all these, except for some variations, SNP variations or whatever variations are there. But there are, they have the similarity. So now what plays here? It plays the insect behavioral genetics. The Rayleigh, which is being reared on the Soria, it has its adaptability of feeding in the Soria, and you are doing this in the adaptability of the feeding in the Arjunanasa. So many variation. So sustaining of the breed will not be there. Once you can make, you can find out, it can succeed for 1, 2, 3, but not for further sustenance. So there should not be like that. And only Daba, we should restrict to Daba breeding. High throughput detection of disease through non-invasive technology. Analysis of nutriophysiogenomics along with nutriometabolic acid identify the protected trait linked biomarker. अब ये वाला पार्ट जो है causes restricting the expression of hybrid vigor. What is it is telling that when we are doing crossing Daba to Daba, the heterosis percentage very small. But when I am doing it with the wild Daba, the very big one, so there is a variation now, cocoon weight. So then it's coming more. So what we have to do? We have to make the different family breed classes based on the cocoon. So that I have made, and on that way we have been succeeded in doing it. You see. Now this term also I was putting, and people were laughing. Why you are telling double laying silk Daba? What is mean? Laying means you know. Laying means you know. Yes. 1 DFL equal to in Tassar 200 eggs. Okay. So 200 eggs in weight is 1 DFL. Okay. But moth are giving more than 200. I have told you that this wild Daba is giving 520 eggs. Wild Daba giving 520, and when it is just bred downstream, it is decreasing 450, 420, 400, 350 like that. So that is why I am telling double laying silk Daba. So silk is also double. The cell weight is also 4.5, whereas in Daba you will find 1.8 or 2.25 like that. But here it is 4. So double laying silk Daba. So one population classes, main class based on fecundity 525, 325, and plus laying, that means plus laying means laying we have taken 200. So any number which is above 200 is plus laying Daba, and average laying silk Daba, that is 225 to 130. Actually 200, but average will come 200. Even some of the moth they will leave 160, 170 like that. In TV, very less fecundity comes because the pupal weight decreases. So in insect breeding or this type of complete crop cycling, there are positive points somewhere, negative points somewhere. You see, Bombyx mori is small, 600 number of eggs. Here, big, but the eggs are large size. And as it increases, very big, eggs are there, but the number increases. So some of the innovative ideas may be created to just increase the fecundity. Why I am telling? In Airy, that is also non-mulberry. Why? Not white because that's completely indoor here. You cannot tell it's wild. It's a semi-domesticated, you can say. The fecundity is 500, 450, 500, just like mulberry. And their generation of some breed is also successful. But in Muga, you are not finding success. Why? I have worked in Muga also 6 years. The same type of Antheria milita, same you will see. There is some variation. The host plant also same. Just like this. Som and swallow. Swallow just like Ashan and Som just like Arjun. You will find there. But there it is multi-voltine. Why? Why it is multi-voltine? Anybody from Northeast? No.
Sir, they have come from Simedi only.
70.
Means after breeding.
Tell. Why? Even though there are so much similarity with the tropical Tassar, but that is multi-voltine. Why?
Sir, they have taken 1 month training, then they have come here. Very good.
You might have learned there. Why? Antheria milita is there, Antheria asama is there.
Actually, there is people diapers in this.
Very good question. But why here people die? Why not there? Why it has broken and why there is merging?
Environmental conditions.
Yeah, very good. Environmental, altitudinal, climatic. You have seen the rainy season starts from April to June, July. Sorry, June, July. October, up to October. Up to October, very long, about 8 months. So you know, the diapause is regulated by altitudinal, climatic, seasonal variation, airflow variation, so many things. So many factors are there for the diapause regulation. So this you can do for the breeding in Daba. And then you can go for recurrent selection on this basis. Intercrossing, composition, and repeating. अच्छा, one I will show you. Yes, it is complete होगा. मैं एक चीज बता रहा हूँ, ये वाला साइकिल को समझ लो, मैं दे दूंगा सर, ले लेना, या हमारा व्हाट्सएप ग्रुप क्रिएट किए हो, व्हाट्सएप ग्रुप, उसका नंबर दे देना, मैं इसमें दे दूंगा. मैं ऐसे यूट्यूबिंग ब्लॉगिंग भी करता हूँ, मेरा फेसबुक में चला जाना, उसमें बहुत सारे ऐसे टीचिंग्स हैं. सो आई विल ऑल्सो पुट देयर सो मैनी न्यू थिंग्स. यहाँ मैं बताना चाह रहा हूँ, ये एरिया देखो, ये जैसे जीरो पेब्रिन के लिए मैं बहुत आई हैव जस्ट बिकम वेरी मच एंशियस फॉर जीरो पेब्रिन, बट इवन दो आई वॉज प्लेस्ड इन P4, I was not finding zero peregrine population. And when I am telling here, scientists were very much awkward that he is telling zero peregrine cannot be there. But there is possibility, zero peregrine. Sal, if a stock is reared on sal, it will, there are so many disease tolerance factors in asan also. If you will ask to the farmer now, they will tell that even dying worms become live in the asan foliage, not on Arjun. Asan foliage has so many of the disease tolerance factor. Just like that, asan is found where? Interspersed among the sal, Soria robusta. Soria robusta का जितना फॉरेस्ट है ना, जितना ट्रैक है, जंगल जब रोड में चलते हो ना, यहाँ से चक्रधरपुर या वहाँ तुम जाते होगे जो नॉर्थ ईस्ट में, बोको वगैरह एरिया. बोको आई वाज प्लेस्ड 3 years, बोको, and 3 years in Golaghat. I have created plantation in Golaghat. Okay, 3 years. So you will find asan interspersed in between the soria. So they have no chemical allelopathy. Allelopathy समझते हो? एक प्लांट दूसरे प्लांट के बीच में नहीं हो सकता है. अर्जुन वहाँ नहीं होगा, but asan will be there. तो जब asan, if asan is there, sal is there, put some DFL on asan and connect the branch of asan to the soria and let the larvae move self, suo moto, don't touch it, suo moto, to go to the sal, and they will breed there, and then you collect it. Means again you just collect it to the asan. So one side you just upstream, another side downstream. So that I have given that in this way you may be able to take some zero peregrine population. Otherwise in P4 there is a restriction, zero peregrine. There should not be any peregrine in the P4 and P3. Even the P2 also. Even only permitted is P1. Now last peregrine वाला बता देते हैं. Now I am coming to that. खत्म हो गया, but I am telling you peregrine, how you have to restrict peregrine. If you are saying the larvae not feeding in the early stage, third, fourth, it is pebrinized. You will see irregular growth of the larvae. It is pebrinized. How you will come to know? Let it to continue. Let it to continue. If it is pebrinized, larvae will be irregular sized. If it will convert into cocoon, pupa will not emerge. It has happened in DOS. I was the committee member, I have went there to deviate the report that it is due to something else. But I was knowing it was due to peregrine only. Moth emergence was completely stopped. पूरा कोकून का मुंह बंद हो गया, इमर्ज ही नहीं किया. This is due to peregrine.
irregular sized moth, this is due to pebrine. So these are some of the symptoms I will share with you. You just give some WhatsApp group number, I will share that. Okay, so this happens. So the pebrine is very dangerous. So eliminate these things. But if it has come in the population, it is very dangerous. So you might have seen some of the photographs. So
Kurjuli photograph is, yeah. This is Kurjuli, the left side photograph. Now this is the cyclic program, but it is Hindi. I can give you. Now this is chromosome number Asama 15, Fahrenheit 49, Proili 49, Yamamai Japanese tasar, Samia Cynthia 14. प्रोडक्शन मलबेरी का कितना है 23-24 में 29,892 मेट्रिक टन, वन है 9,021 जिसमें तसर है 1586 और एरी है ज्यादा, एरी इज मोर 703 मेट्रिक टन एंड 252. So what is the best thing? Host plant is important, pebrine zero is important, person farmers important, climatic stresses. Thank you.